Hello everyone, my name is René Diaz and I'm going to be talking about the chemical 1,4-DMN and treatment on potato seeds. First off, on the research paper, the three types of seeds that they used were the russet Burbank, the Umatilla russet, and the Ranger russet. Now these types of potato seeds were used because they're the most commonly used on companies and industries. They're typically used for French fries or even potato chips. Now, they use the 1,4-DMN chemical, which is dimethylnaphthalene, because it's a chemical that is actually expressed by potatoes whenever they're in a dormant state. Now, scientists have figured out a way to make this chemical synthetically and then apply it again to potatoes that do not express this chemical in order to keep them in dormancy. Now, the good thing about keeping a potato in dormancy is so that they don't sprout. Like you see here in this picture above, if a potato seed sprouts, then it means that it's not edible. Therefore, the only thing you can use it for is for farming. Now, if you have a dormant state potato, it can still be used to be eaten or to be edible. Therefore, companies or industries, whenever they have their whole potatoes locked up in a warehouse somewhere, they want to use the DMN to keep them in a dormant state so they don't sprout. Because if they sprout, then that means that they lose money because they can't use that potato on their products. Therefore, we're using this DMN to prevent the sprouting of potatoes, which means to keep it in a dormant state. Now, this research paper is looking at how productive would a potato be, even though if it's on a dormant state, whenever you farm it. Now, the point is, or the difference is that a potato that's already sprouted will have a higher, not a higher rate, but it will be fast to harvest or to grow. Now, the potatoes that are in dormancy state will take longer to sprout and grow because they have to leave that dormant state, which means the DMN chemical has to degrade and let the potatoes sprout. Now, the experiment starts with a time span of 2 years. Now, they lock up the potatoes for 2 years in different temperatures to get a degree day data on the sit potatoes. Now, they found out that actually, like I said before, the DMN potatoes did actually had a delay whenever they were in the process of farming, rather than the control group, because the control group had already sprouted. Therefore, they'll have a faster rate, and they'll have a faster rate to reach to productivity. Now, even though DMN potatoes had a slight delay on their rate, they still reached to maximum productivity, which means that the DMN does not affect the productivity of potatoes, just rather delays it. The russet Burbank was the first one, or is the first one, to have the highest productivity from all potato seeds. The second seed, which is the Ranger russet, has the second highest degree of productivity, and the Umatilla russet seed has the third highest degree of productivity, in which you can see here that both control and DMN took time to reach the maximum rate of productivity. But nonetheless, this DMN does not affect the productivity of the potato and does not affect how the potato farming and the harvest of the seeds. It just keeps it in dormancy so it will still be edible for a longer period of time. And that's good for companies because it saves them money and they can store these potatoes in the warehouse for a longer period of time without having the potatoes to spoil. Now, I go back to the potato to spoil. It was interesting because even though the DMN was used to prevent sprouting, the potatoes also had like less chances of being spoiled by fungi and bacteria. So Dr. Campbell came to me and Ravi and he said, hey, we think that DMN also has an effect on the potatoes, inhibiting the growth of bacteria. So our research here in Barron is that we proposed that maybe this DMN, dimethyl naphthalene, could work as a preservative. So the other question is it safe compared to the current chemicals that are being used by companies? For example, CPIC. CPIC is a synthetic chemical that's also used now on the potatoes to prevent the sprouting. Now, we suggest that we could use the DMN, the 1,4-DMN, because it's a more natural chemical, which is actually expressed naturally by potatoes, rather than use a synthetic, unnatural chemical on the potatoes, which have some side effects or even change the structure of the potato. Actually, not that much, but it's still a synthetic. So we'd rather use the DMN because it's a more natural chemical, because it actually resembles, even though it's synthetically made in labs, it still resembles the same chemical structure than what the potato express. So on our experiment, we did 32 samples. We did 5 treatments, and each treatment have 32 samples. So we did 2 types of samples. We did a DMN and a control sample. Now, every time we take a sample, it was a nickel-sized sample from the potatoes. So we took 8 samples from DMN, which was the skin of the potato and the inside of the potato, and the same goes to control. We did this because we want to compare the samples from DMN with the control and see what bacteria grows on the control and to see if they do not grow on the DMN, and that includes for fungi. Now, we use 10 microliters, excuse me, we used 10 microliters of the DMN and water. And what we did is that we put it in this capsule, and then in this capsule had little capsules that we put the strip in. So we close that, and then they'll be concealed airtight in here so the chemical will treat the potatoes. Now the control had distilled water, and the DMN had the DMN chemical. And I put here a picture of the structure of the 1,4-DMN. Thought it would be interesting. Now, after treatment, we isolated the DNA of the potatoes. We technically used the nanospec to actually see if we had DNA in our samples, which I think was cool. It was the first time I used it. This is just a picture of some of the samples that we had. After the DNA isolation, we used the PCR tubes. Now the PCR tubes, it locates the 16S RNA. So the PCR is wherever we put the primer in to identify the different types of bacteria. Now we did 3 different types. One was for V3, which is a specific types of bacteria. The second one was V4, which is another specific types of bacteria. And the last one was fungi. So we have 3 different sets for the same samples. After the PCR, we took the gel analysis. And you can see here that this actually looks pretty nice. They all look pretty nice, which suggests that we did actually isolated the DNA and the good PCR samples. So we send that to UP so they can sequence it, and then they brought back the sequences to us. Right now, we are currently using the mother program to actually identify and analyze all the sequences that we got back from mother. So we're currently constructing our abstract. We still haven't finished analyzing it, but we hope to finish by this month, or maybe by the beginning or before spring break. We have high hopes that we actually find good research results. I had to give acknowledgement to Dr. Campbell, which was who gave me the opportunity for this research. Olivia, which helped us during the summer with the samples. Ravi, which was my research partner, and the 1-4 group Meridian, which were the group that funded this research. Thank you.